Dear students, and this is the class for class 11 biology, CBSE and also ISC. Today we are starting the chapter biological classification. So let us write the chapter's name, bio biological classification. Okay, so the whole living world has to be classified. As you know that whatever you have to give in a proper way. For example, in a school, from class 1 to class 12, all the students cannot be kept in one room and giving the all the teachings. It is not possible. So what has been done? The classes, they were divided into, the students were divided into different classes from 1 to 12 according to their brain capacity. The criteria is brain capacity. Similarly, the living world is divided on the basis of different criteria, the whole living world. And it was not divided on one or in a very short time. It was divided after great research by the scientists or taxonomists, and gradually it was divided into different kingdoms. The first kingdom was very gross, that is a two kingdom classification. Okay, the first one we can say that is the two two kingdom classification, that is the animal kingdom and plant kingdom. So you know that now you very much know that this is a gross classification. Now second classification was done, three kingdom classification, three kingdom classification. And in two kingdom, what I had said, animal kingdom, plant kingdom. Okay, so it was grossly divided into animal kingdom and plant kingdom. And now we will go to the three kingdom classification, and there they have written what it is. Plantae, and plant kingdom can also be called as Plantae, Animalae, and then came the Protista. Okay, then came the Protista. Okay, so what this Protista word is very new to you, and Protista means what? The organisms which are unicellular, made up of only one cell, those are called protista. That's all. So the scientist came, just did the research work and found some unicellular organisms are mixed with plant kingdom and animal kingdom. It has been derived separately and then kept in the kingdom protista. Okay, so next, that time they did not bother about the development of the nucleus. The scientists did not bother about the development of the nucleus. Later on, it was again focused by the scientists that okay, unicellular is okay, but some cells have well developed nucleus and some cells have not well developed nucleus. Then how can they kept in one kingdom protista? So again the division came, that is the four kingdom classification. Okay, children, that is the four kingdom classification. So how will you now memorize? It is very easy, like a memory game. You just write all the first one, that is the whatever you have written first, that is the Plantae, Animalae. Okay, let it be like this. Then Protista, and then you write the new one. New one means Monera. Okay, so Monera, they just derived it on the basis of the nucleus, that is the prokaryon, prokaryotic cell. Okay, you have the knowledge of prokaryotic cell. Now, then again the scientists found that In plantae, there are some organisms which cannot photosynthesize. And how can they be kept in plantae? They cannot be kept in plantae, so they were separated. So again, the next classification type came, that is the five kingdom classification. Five kingdom classification. Again, the memory game. The first friend told you plantae, so you have to say plantae. Next friend told you animalia, so you have to say animalia. You have to remember and say animalia. Then the next friend had told protista, you have to remember and say protista. Then the monera. And what about now? This is the fungi, fungus, which were first, fungi is the plural, fungus is singular, which were first kept in plantae, but they could not photosynthesize. They were separated. So now this is the five kingdom classification, and it is, you just mark it as star, because the question will come in your class on this five kingdom classification, because all the kingdoms now we will study on the basis of these five kingdoms. So the scientist name is also very important, and so you can write the scientist name as R.H. Whitaker. Scientist name, others also you will get in the note, but here this is important. So the scientist name I am giving as R.H. Whitaker. Whitaker. R.H. Whitaker, and this is the year 1969. Okay, so this is the just a summary of the history, and I had mentioned what are the demerits of two kingdom, why three kingdom came, why four kingdom, five. So I have mentioned the all you will get in the notes which I had already given. So now we will proceed to the next topic, that is the criteria. If we are considering the five kingdom classification, what are the criteria the scientists had undertaken, that you will get the question in your school exam. It will be of 1 or 2 marks. So let us now see that. Okay, so now clear. So now we are going to the criteria. What should be the criteria? Criteria will be morphological. Criteria will be the nutrition pattern. Okay, so let us read the criteria according to the classification. History of the classification. How? See, first, Monera protista. These were derived on the basis of what? These were derived on the basis of development of the nucleus. That means the structure of the cell was given importance. So one of the criteria is the structure of the cell. Next, that was the this Monera protista unicellular, and then the unicellular organ. Monera protista were separated from plant kingdom and animal kingdom because let me select this one. Yeah. So, yeah. So, Monera protista, they were separated from the plant and animal kingdom because they were made up of only one cell. So, the criteria first, what we will write the criteria. Criteria for classification. Criteria for classification. Okay. So, what are the criteria? First, the structure of the cell, means the nucleus, structure of the cell. You can see the structure of the cell and what is the type of nucleus. So this will be one criteria. Like this, the Monera protista had been done. Then we will see the number of the cell. Means we will say this one as this body organization. We will say this one as body organization. Okay, body organization. ZATION or SAION, body organization. According to that, here that is the prokaryote and this eukaryote was been done, and your body organization, it will be unicellular multicellular, number of the cell. Okay, body organization, number of the cell, or cellular or tissue level, or what, that is also under body organization. Next, we this plant, from the plant, the fungi were derived on the basis of the type of nutrition. Type of nutrition, that is the autotrophic and heterotrophic. These green plants were kept under plant kingdom. Fungi were derived or separated from there because fungi could not make their own food. So this is also a criteria. Okay. Next, it will be what? This body structure of the cell, body organism, type of nutrition, and then phylogenetic relation. Phylogenetic relation means what the relation they have with the previous generation, what the relation they have with the previous generation, that is the phylogenetic relation. Okay, relationship. Okay, so here this is phylogenetic relation, that I will explain now because this is a new term, phylogenetic relation. And number 5, the reproduction. What is taken consideration? Reproduction. The type of reproduction, vegetative or what. Now this is a new term, phylogenetic, means the evolutionary basis. The best example I am giving, under the vertebrate, there are many classes and it starts with pieces. There is a fish, then it is amphibia, then the reptilia, then the apes, and then the mammalia. If you will see here in this class 11, you will see that the structure of the heart in class 10 you have read, fish has only 2 chambers. So the mixed blood circulates through the heart. So it has 2 chambers, fish, and then the amphibians have 3 chambers. Okay, reptilia also have 3 chambers, but in crocodile you will get 3.5 chamber, and then a mammalia 4 chamber. So is it not a evolution, gradual progress from 2 chamber of fish to the amphibian, that is a frog 3, then the reptilia in some species, that is a 3.5, and then mammals 4. So this gradual progress has a link with the previous generation, previous class. This is called phylogenetic relationship. Over, my dear students. Can you understand? Yes, ma'am, we understood. Clear? So, criteria we have done. So, let us now proceed to the next topic. That is a very interesting topic. We will see, that is the kingdom Monera. The kingdom Monera. Okay. So now we are starting the 5 kingdom classification, and I must say there is another classification, that is a 6 kingdom classification. All the 5 kingdom you have to write, and a new kingdom the scientists had derived, that is the archaebacteria. As it is bacteria, so it was included in Monera. Now I will start Monera. Then you will understand what is there under Monera. Then you will understand what is Archaebacteria. So in Monera only Archaebacteria was actually included. Okay, but the latest research work derived the Archaebacteria from the Monera and made a 6 kingdom classification. Why it was separated? Because of its unique characteristic feature of Archaebacteria, which the other normal eubacteria do not have. And what is the unique feature of Archaebacteria? They can resist high salinity, high temperature. Okay, so this is not possible for a bacteria. For example, temperature in your daily household activities, when there was no filtration process of the water, drinking water, what we used to do, there is a boiling of water. Now also, for safest process is boiling of water. Why? Because in boiling, higher temperature will kill all the microbes, including the bacteria. But archaebacteria you cannot kill. Even there is another type of microbe, actually you will read in this chapter, that also cannot be killed by high temperature. So why? Because of very special feature of the cell wall of archaebacteria. Okay, so that we will read in now, as we are following the Whitaker's five kingdom classification. So we will read Archaebacteria in five kingdom classification, that is under the Monera. So let us write Monera. Okay. First, the characteristic features, general characteristics characteristic features. I think it is very much visible. So first, you know, that is cell type. If you are asked what is the cell type, you will say prokaryotic. Prokaryotic. That is the ill-developed nucleus, primitive nucleus having no nuclear membrane and other parts of nucleus. It is only the genetic material floating on the cytoplasm, having no barrier, having no membrane. Okay, no separation between the genetical material and the cytoplasm. So there is a primitive type of nucleus that is called the prokaryotic. Okay, then the if you are asked body organization, then you will say that cellular, that is cellular. Okay, number of cells mostly unicellular, but some filamentous bacteria are multicellular. Okay, this is the okay, clear. Then we will find what are the organisms. Here I am coming. Okay, so let us this. What are the organisms? Organisms which are found here, that is the bacteria. Yes. Note it down if you are noting down. So these are the general characteristic features of the kingdom Monera. Okay. I have given you a little time. So now let us move ahead. That is the how we are classifying Monera. Okay. So let us write Monera. Monera. It is divided into, according to 5 kingdom classification, it is divided into 2. That is the Archaebacteria and then the Eubacteria. Okay. Now, is it okay? Archaebacteria, Eubacteria. Then again we will classify this. Again we will classify this into, clear? The characters which are in Eubacteria we can classify. You know this very well, this classification. First, first, first we are classifying this according to shape. So I am writing here shape. What is the criteria I am taking here? Shape. Clear? I am taking here shape. You can, is it visible? Shape. Now again choosing white. So according to shape, number 1 is Kokki. That is the plural one, and their shape is spherical. Okay. Number 2. Number 2 is Bacilli, and their shape is rod shaped. Number 3 is Vibrio, and their shape is comma shaped. And next is Spirilla. 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 Is it visible? Spirilla. Their shape is spiral. Okay, spiral. Yes, students, this I'm sweating very much. So they are according to spherical, bacilli, vibrio, spirilla. Now we will write the example for each. Streptococcus, Escherichia, spirillum, vibrio, choleri. Acha. Okay. So example I am writing with another pen. Let us write with red. Is the red visible? You tell me. That is the Kokki. So example I am writing here. Sorry. Okay. So Kokki. Now the example with red pen I am writing. Strepto. Strepto. This is example. Streptococcus. It is genus. So underline, then species. So this is an example. Streptococcus. Okay. Now bacilli, that is a rod shaped. What you will write? Escherichia. This example is very important for class 12 also. Escherichia. More example are there, but we are just giving one here. And then it is the Vibrio choleri. 1 minute. Okay. Vibrio, comma shaped. The example is Vibrio. Vibrio. Underline Vibrio choleri. You will get all this in my notes already available with other students. Those who have missed the class, they will take the notes. I cannot write the whole note on the board because it will take too much time. So I will give important parts only on the board. So spirilla, that is the spirillum. Example is very easy. Example is very easy. That is the spirillum. This is the genus and this is the species. Okay. So this is according to the shape. Now let us take another criteria, that is according to the nutrition. We are taking the criteria, this blue pen we had selected, and here we are writing nutrition. So we are taking the criteria nutrition. So in that, we will write with again the white one. So we will write number one, that is autotrophic. Okay, autotrophic. Okay, and number two, that is the heterotrophic. That again the division is there that you will get in the notes. As I said, the whole I cannot write on the board now. So autotrophic means autotrophic, they can prepare, these bacteria can prepare their own food, and like the plants, some bacteria use solar energy, solar light as the source of the energy, and they are phototropic. And some bacteria use some chemicals as the source for the energy, they are chemoautotropic. Phototropic, chemoautotropic. Okay. And under heterotrophic, you will get mostly the parasites. And you know, when the bacteria are the parasite, mostly they are pathogenic. So disease causing bacteria are included in heterotrophic. And in case of uh, heterotrophic, you will get another type of bacteria. It is a parasite I had discussed as the pathogenic in the host. And another will be like saprotropic, and which are getting nutrition from the dead and decayed like fungus. Okay. And another is the symbiotic relationship. They are mutually related to another organism, and from there they are getting the nutrition. Okay, so this is the that is the division on the basis of nutrition. Now there will be another criteria, that is the requirement of oxygen. That is also important. What I said, that is the requirement. So requirement, there should be synchronization, and I like that. Representation is very important in your exam also. So I should write here requirement of O2, and according to that, we will be having two. One is uh, one is this, that is the aerobic, and another will be an aerobic. Okay. So these are the, this is a, you can say that grossly we have divided the bacteria in this way for the ease of study. Okay. So now we are going taking the another topic, that is the archaebacteria. So students, how are you liking? This is bacteria. Okay. They are so small, microscopic organism, but that also you have to study because it will come in exam. So let us see the archaebacteria. I think everything is visible. You have noted down. Giving you 1 minute. Okay. So I think you have noted down everything. Now let us see what is archaebacteria. So now our topic is archaebacteria. Okay. Let us listen. You will get everything in notes. So listen. What is there? Archaebacteria, actually a special type of bacteria which can resist, as I said, high temperature, high salinity. Okay. And this is due to their special structure, that is a cell wall structure. We know that the bacteria, they have a cell wall, and that is made up of peptidoglycan, then cell membrane and everything, whatever you have studied about the bacteria. So here the cell wall is not made up of peptidoglycan, it is made up of pseudopeptidoglycan and glycoproteins. Okay, and the cell membrane is having only a single lipid layer. So all these internal structure, cellular structure, make archaebacteria to survive in extreme conditions, which a normal bacteria cannot survive. Okay, so let us now write this one, that first of all, unique feature only I am giving you here, others you will get there. That unique feature of archaebacteria is life. Okay, okay, so keyword is survive extreme conditions. Okay, so this is possible. Unique chemical structure of cell wall and what I said, and cell membrane. Okay, so this, what is the chemical structure you will get in the notes? It is made up of pseudopeptidoglycan, polysaccharides, and this glycoproteins. And here cell membrane, single layer of lipid. These proteins actually make them stable in the adverse condition. Is it clear? So this is one of the very unique I discussed. Now we will classify the archaebacteria into three categories, okay, according to their habitat. I think you have not written, I am giving you 1 second, 2 seconds to write it. Have you written? I am just giving you the time to write. I think you have. Now let us start. the classification. Archaebacteria, oh sorry, the criteria taken as habitat. Criteria taken as habitat. Habitat means a dwelling place, and then they are divided into halophils. 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 You can see halophils. Then methanogens. And then thermo thermoacidophils. Thermoacidophils. Okay. Now, what are these halophils? On habitat is what in halophils? This question will come in your mind. That is the high salinity. High salinity. There. Okay. So in high saline condition, these archaebacteria will survive. These methanogens, marshy areas, okay. Then in in ruminants, you know, ruminants, the cattle, those who once eat some food and again they bring it in their mouth. Okay, so these are, and they have a special largest chamber in the gut, that is the rumen. So in ruminants, okay. And thermoacidophilus, as you can see from the name, thermo mane temperature, and so it is high temperature. High temperature. So in this way they have been divided. So we are studying for board, means class 11 school exam. 11 will not come in board. So on the basis of NCERT, CBSE and one reference book we are studying. But for NEET you have to study details of this. Here we are not going in details. Clear? So this I will say the high temperature, this will be necessary to recall in class 12 when we will study the PCR, that is polymerase chain reaction in the chapter biotechnology. There you will get one thermoacidophilus archaebacteria, that is the Thermus aquaticus, which can survive in high temperature, and the enzyme is the enzyme for PCR. PCR is actually conducted under very, very high temperature, and the enzyme for that PCR is now derived from this archaebacteria Thermus aquaticus. So this is important for class 12. And their methanogens, they are important because they release the gas, that is the methane, which is again a very important gas that is release methane. So the name is methanogens. Here the sewage treatment in class 12. Again you will get this one in anaerobic, some anaerobic bacteria, they release the gas from there in the third tertiary treatment of sewage in class 12. Okay. So here they release methane. How do they release methane? They actually remain in the rumen, that is the gut of some ruminants, and they help to digest cellulose with other some bacteria, that is through the fermentation process. In that process they release methane. One way, this one. Another way, from the dung, cow dung, they release methane, these bacteria. So it is given in the note, the process in the flow chart, very short small flow chart. You can go through that and you will get it. So write it down. Okay. Now we will proceed. So now we are going to the eubacteria, Archaebacteria, eubacteria also. I am giving in a summary. You will get everything in notes. Now let us see the important points in eubacteria. Eubacteria, the cell wall is made up of peptidoglycan. Cell membrane has the lipid bilayer, ester bonds and all. Okay. So in eubacteria, you will get first the cyanobacteria. That is a cyanobacteria. Very important, cyanobacteria. Here, other points you will get in the note, but the point which will come in school exam, that is cyanobacteria can directly fix nitrogen. Directly fix nitrogen from the atmosphere. Which plants also cannot fix. That is, animals also cannot fix, but they can fix. How? There are some example we will see. That is the nostoc, anabaena. These are all genus. They can fix. Cyanobacteria, they can fix. Okay. So how do they fix? I will show you. This is important for school exam. And so let us draw this. Let us select this one. And I'm drawing the filamentous nostoc. Okay. So let us first do this like this. This is a filamentous form. And here I am drawing the cells. As I had said, that is the filamentous form. Okay. And this is not okay. Let us draw properly. This I had selected. This is the filamentous form of nostoc. Okay. Nostoc. And if we will see the cells like this, this is the cells made up of these cells. Here also. These are cells. And here I will make with another color. Then you can see the difference. That is a cell like this. A cell like this. First let us draw and then we can draw this one. That is a cell, normal cell as it is where. Yeah. And This will be like this. So in this board, it is difficult because this coming like this eraser and all pen. See, again it is erased. Shit. So what I meant to say, just let me complete. That is, let me complete, then you see. Now see, this one, the red one, that is a bigger cell, okay? Bigger. Okay, first bigger, then thick walled, okay? Thick walled. And this is having anaerobic condition. Here, anaerobic condition is maintained. Everything I will explain now. Anaerobic condition is maintained. So this typical cell fixes the nitrogen. This fixes the nitrogen, fix N2. But other cells, these other cells, they are just vegetative cells. They are just vegetative cells. They do not fix nitrogen. Okay. So if the question comes, the vegetative cells cannot fix nitrogen, but this bigger, thick walled anaerobic condition cells can fix nitrogen, and they have a specific name, that is the heterocyst. What is the name? Heterocyst. Okay. Different, hetero means different, is different from other cells, and cyst, it is having a very thick wall. It is heterocyst. And why it has this uh, anaerobic condition? Because it fixes the nitrogen by the help of the enzyme nitrogenase, which will be destroyed by oxygen if oxygen will be there. Okay, so it maintains an anaerobic condition. And how does it get energy? It gets all the energy through oxidation from the vegetative cells. Okay, you can say that inside the body of the nostoc, you can see a symbiotic relationship. That is, the other cells are getting nitrogen from this heterocyst, and heterocyst is getting energy from the vegetative cell. So it is very important for class 11 school exam, that is a nitrogen fixation. Okay, nostoc. Okay, now why this cyanobacteria is also called what? Blue green algae. Blue green algae. Why? Because of some pigment like phycoerythrin, phyco, this green color is for chlorophyll A. ठीक है? Okay, and this bluish grayish color, that is a phycoerythrin, phycocyanin. All this phycocyanin contains copper. Copper gives blue color. Okay, so blue color is for that, green color is for chlorophyll. So all these pigments are present in cyanobacteria, so they are called blue green algae. Clear? So you can write this. Give me some time. হ্যাঁ, বলছি যে আমি তো এত লং দাঁড়িয়ে পড়াতে পারছি না, এবার তোমায় বসতে হবে, বাট বসলে তো আমাকে দেখা যাবে না বোধহয়। কি? ওটা মানে সাইডে চলে যাবো তো, স্টপ করার কিছু নেই। আচ্ছা। ওকে, ওকে। হুম, ওকে। ঠিক আছে, ঠিক আছে। হুম, হুম। Okay, students. So up to this, we had studied Eubacter cyanobacteria. Now we will go for these are all autotrophic bacteria. That is the photoautotropic, chemoautotrophic, and now we will move to the heterotrophic bacteria. As I said, the heterotrophic bacteria will be like this decomposers. Okay, and those are very important bacteria because they help to clean the environment. Decomposers, and then this. I said about the pathogenic bacteria. They cause some diseases, and the diseases, some of the important diseases which will be coming in your school exam, that I have given in the notes with the pathogen, name of the pathogen. You have to memorize that. And then some will be for harm, for beneficial, like the making the curd from the milk, that is a lactobacillus, making the curd from the milk. Okay, some like this, the bacteria, and as I said, that is the heterotrophic bacteria. They can be parasites, symbiont, saprotropic, and some helpful like lactobacillus and all. So these all names and all you will get from the notes. Here I am not going to write all those names which will linger the process. Okay, now we will go to the reproduction in bacteria. So bacteria also reproduce, and you know the bacteria is under monera, very primitive organism. So they cannot reproduce like the higher animals and plants through the sexual reproduction. They, if the question comes generally in school exam, it is asked that how do they actually reproduce. That is a vegetative form, that is asexual form, that is binary fission. Binary fission, you are acquainted with binary fission because you have read amoeba in class 10. So similarly, bi means two and fission means breakage. So the whole cell breaks into two in case of binary fission, and first the cell becomes elongated, and then the cytoplasm also gets with the cell. Definitely it will extend, and then the cell membrane moves inward, and the septum forms in between. Septum forms, and this septum divides the single cell into two. Okay, with the division of the genetic material, that is the not nucleus, that is the genetic material replication. The genetic material, that is a DNA, if we consider genetic material is a DNA, that will replicate into two. So one will come in one cell, another will come to another cell. Okay, and this genetic material is also called the nucleoid. It is made up of only a single chromosome and floating in the cytoplasm. All about the reproduction, and the another type of reproduction is also observed in bacteria, where the two different means the bacteria, if the bacteria is there with its own DNA, it can take DNA from other through conjugation tube or through some pili connection. So that is the mixing of two DNA. Bacteria A has its own DNA. It is taking the DNA from other, like this. Suppose the bacteria is attacked by the bacteriophage. Then the bacteriophage is a virus which is having also genetic material. If it is attacking or coming into the host, so it will transfer to the. It will take the machinery of the bacteria's DNA. So there will the mixing of the genetic material like virus and bacteria. So it is a mixing combination. So you can consider there is a variation. So it is not sexual reproduction, it is just only recombination. Recombination means combination of two different genetic material. So let us write this one into a very concise way, that is reproduction in bacteria. The elaborate notes or explanation, which I have just verbally told you all, we will get in the note. And here the okay. So let us take a break of 5 to 6 minutes. Then we will meet again. Okay, students, I am back. Now, the last part, today's last part, I am going to discuss, and that is the very interesting. There's a mycoplasm. Okay, so mycoplasma, I will give some points, and that is the end of today's class. First is the smallest organism. These are smallest organisms, microorganism. Lack cell wall, so cannot be killed by antibiotics. Why? Because antibiotics target cell wall, so it has no cell wall. Can survive in anaerobic condition. Conditions, so can change shape, has no cell wall, and the last point is the pathogenic. Pathogenic, they can pathogenic. Okay, so they can uh, uh, eh, cause disease. They can cause disease. Disease name is given in the notes. Go through the note. So it is a very interesting, important topic for school class 11 school exam. So bye students. Tata. Please go through the notes and take the notes. Thank you.